The next type of cell division is called meiosis. Now it differs from the mitosis in that meiosis is called reduction division. While mitosis is a cell division that is required for cells growth, like growth of the stems, growth of new branches, or in case of animals, the skin for skin repairing, to grow the number of cells, to increase the number of cells and help in growth. That's what mitosis is for, whereas meiosis is for producing gametes. In mitosis, the chromosome number remains the same. Chromosome number of parent cell is same as the chromosome number found in the daughter cells. Whereas meiosis is called as reduction division. Meiosis is called reduction division because the chromosome number reduces to half. Chromosome number in the daughter cells is half, is reduced to half. Half of what is found inside the parent cell. So that's why they are called as reduction division, and they are used to make gametes like sperms or egg. Now, meiosis can be divided into two stages. One is meiosis 1 and meiosis 2, and each stages has their own subphases like prophase 1, metaphase 1, anaphase 1, telophase 1. And meiosis 2 is divided into prophase 2 and metaphase 2, anaphase 2, and telophase 2. Prophase 1 stage. So during the prophase 1 stage, you can see here the chromosomes that have almost the same length and they look similar, that is called as homologous chromosomes. They found attached. So prophase 1 can be divided into various subphases. The first stage is called leptotein stage. Now, during the leptotein stage, the chromosomes will start to be visible because during the interphase stage, they were not visible in this form. They are now visible. The chromosomes are visible in the nucleus. That's what is there in the leptotein stage. Next is zygotine stage. In the zygotine stage, there are pairing of the homologous chromosomes. That is the chromosomes that looks similar in their length and sizes. Now these two are homologous chromosomes. So in zygotine stage, we can see the pairing of chromosomes taking place. Stage is called Pachytene stage. During the Pachytene stage, there will be crossing over of the different segments of the chromosome. Crossing over takes place. And for the crossing over, there is an enzyme also involved which is called recombinase enzymes. So you can see here there is a crossing over between the segments of non-sister chromatids. Like if you take a look at the chromosomes, these both are sister chromatids. These are also sister chromatids. Both the chromatids attached to one centromere, they are sister chromatids. But the crossing over takes place between the non-sister chromatids that are homologous. So that is what is taking place here. There is an exchange of the gene segments which is called crossing over. And the crossing over takes place between the non-sister chromatids. The next is diploid stage. During the diploid stage, you can see that the complex formed as a result of crossing over between non-sister chromatids. This complex is called as synaptonemal complex. And this complex slowly starts to dissolve away. So the dissolving away is clearly seen in the diploid stage. So in the diploid stage, when the synaptonemal complex is dissolving away or separating away from each other, there's a tendency to separate away from each other. So this forms the shape of a letter like X. So we can see that the synaptonemal complex dissolves away. They are dissolving, forming X shapes. And so this structure which is formed, that is X shape, is called as chiasmata. And then the last stage of prophase 1 is diakinesis. Now in diakinesis, there is complete separation. There is separation of chiasmata region And we can also see the recombined segments. Slowly, the nuclear membrane also disappears. The nuclear membrane, as well as the nucleolus, is also not found. They both disappear. So here in the diakinesis stage, you can see that the chiasmatas has separated, and we can also see the recombinant segments of the non-sister chromatids. Like here you can see this homologous chromosome has received one segment from the first homologous chromosome. Similarly, the first one has received the segment over here from the second chromosome. So the exchange has taken place. So this is a very important characteristic of sexually reproducing organisms, where there is genetic segments exchange, recombination, and that is a result of the variation found in sexually reproducing organisms. Metaphase 1 stage, the homologous chromosomes are attached at the equatorial plate. So here you can see the equatorial plate, and that is where the homologous chromosomes are attached. So these are the homologous chromosomes. Now these homologous chromosomes, we can also see the exchange gene segments on some of the sister chromatids after the prophase stage. And centrioles that produces microtubules are also found where the microtubules are attached to the kinetochore region of the chromosomes. So this is a kinetochore that is found near the centromere. So microtubules from the centrioles are attached to these kinetochores. Now the next is Anaphase stage. During the Anaphase 1 phase, whatever homologous chromosomes are arranged at the equatorial plate, they separate and they are being pulled towards the pole area by the microtubules that's produced by the centrioles. Chromosomes were attached during the metaphase 1 stage. These homologous chromosomes will be separated towards both the poles. The next is telophase 1 stage. During the telophase 1 stage, nuclear membrane comes around these separated homologous chromosomes and nucleolus also will reappear. The nuclear membrane appears around the separated homologous chromosomes and nucleolus also will reappear. So with this, the karyokinesis is over. Karyokinesis or the nuclear division for the meiosis 1 stage is completed and followed by the karyokinesis is cell division, that is cytokinesis. So this cell will divide into two. Next is meiosis 2. So meiosis 2 is like somewhat like mitosis itself, and it starts with prophase 2. It starts with prophase 2. The prophase 2 stage, at the end of the prophase 2 stage, we can see the nuclear membrane has disappeared and the nucleolus also has disappeared. We can see the chromosomes condensed state. The chromosomes are arranged at the equatorial plate, and the spindle fibers from the centriole attaches to the kinetochore area of the centromere. After metaphase comes the anaphase stage, anaphase 2. So the anaphase stage, you can see the sister chromatids have got separated from the centromere area. So sister chromatids have become pulled towards the poles. The last stage is telophase 2. So during the telophase 2 stage, the nuclear membrane will reappear around the cystochromatids. And the nucleolus. Both the nuclear membrane and nucleolus will reappear. Followed by the telophase 2 stage is the cytokinesis, where these two cells will now divide to form four cells and they are haploid. Where in each nuclei, in each nucleus, where in each nucleus we can see two chromosomes, whereas a parent cell had four chromosomes. So the chromosome number gets divided into half, and that is the significance of meiosis. Then you will get the four cells, and each of the cells have haploid number of chromosomes. Of these haploid number of chromosomes, we can see there are recombinants with the exchange gene segments, and there are also chromosomes that are of parental types. And these recombinants leads to variation in sexually reproducing organisms. Now, this kind of division, that is meiosis, is carried on for reproducing cells like gametes. So, for the formation of gametes, meiosis is meiosis. For the formation of gametes, meiosis take place. So, cytokinesis leads to the division of cells, leading to four haploid cells. So each of the nucleus, they have only haploid number of chromosomes when compared to the parent cell. Like the parent cell had four chromosomes, but here you can see only two chromosomes, which means that if the cell is diploid, then after the meiosis, the cell will become haploid. And this kind of division, that is meiosis, it leads to variation, and that's the significance because of the exchanged gene segments during the prophase stage. And this meiosis is used for producing gametes in sexually reproducing organisms.